Well, it looks like our numbers are starting to stabilize. I think most everybody's here. Thanks everybody for coming out tonight. My name is Jeff Turk, and I'm the chair of the Department of Chemistry at Alma College. And today we're going to be watching a presentation that's a part of the Your Health Lecture Series. This is a collaborative community sponsored by Michigan State University College of Human Medicine, and it's partnered throughout the state. This particular event is hosted by Alma College. Maybe you've seen multiple invites there throughout the past months. We've had co-sponsored by Alma College, Mid-Michigan Health, and MSU College of Human Medicine. Today's speaker is Dr. Sascha Drewlo. He's received his PhD from German Sport University in Cologne, Germany in 2006. And he did his postdoctoral training at the Lilienfeld-Tannenbaum Research Institute in the University of Toronto, Canada. Prior to joining MSU, Dr. Drewlo was associate professor in the Department of Obstetrics, Gynecology, and Reproductive Biology at Wayne State University from 2016 to 2017. He's currently an associate professor in the Department of Obstetrics, Gynecology, and Reproductive Biology in the College of Human Medicine at the Michigan State University. The goal of his research is to close the gap between the diagnosis of placental dysfunction and its treatment. He has interests in the early detection of fetal and placental disorders, along with deciphering the underlying molecular mechanisms of abnormal placentation to ultimately develop novel intervention strategies for preeclampsia, as well as preterm birth and intrauterine growth restriction. So with that, I'm going to turn it over to Dr. Drewlo. The floor is yours.
Thanks, Dr. Turk. Thank you very much for the kind introduction and inviting me today to tell you a little bit about our research and the work we are doing here at Michigan State and beyond. So before I start my talk, today we talk about imagine every baby has a healthy start. I'm gonna give you a little bit insight on what is going on in research and beyond. I have a little disclaimer as well, as it is appropriate for such a lecture as we're reaching out. So I'm not providing any clinical advice to you today, but I might give you insights in what is going on in pregnancy and beyond. And so if you have any questions, please ask your healthcare provider. I have as well, as part of the research, several patents and hold grants regarding placental development and the use of cells from the placenta for the purpose of perinatal diagnosis and commercialization. I'm as well a co-founder of and significant shareholder of a biotech company in San Diego, Cradle Genomics, which licensed part of the technology I'm presenting today. But I'm not directly involved in the operation of Cradle Genomics. All the statements here today are MSU affiliated research communications, but not on behalf or affiliated with the company. It's a little bit verbiage, but it's important, I think, to be very transparent. So today I want to talk to you a little bit about pregnancy, the placenta, and its health, this relationship to health for the mother and the child. I want to talk about possible problems with the placenta during pregnancy and its management, and why the placenta is so amazing. I'm going to talk about, as it relates to my disclosure earlier, in part about novel strategies and research to detect and improve pregnancy outcomes very earlier as ever done before. As Dr. Turk alluded a little bit, our mission really is to enable families to have the best pregnancy outcome possible. Our vision is, as he mentioned as well, to close the gap which is currently there for early detection and the treatment of pregnancy at risk by developing cutting-edge tools and intervention strategies. As you probably all know, pregnancy is wonderful, but it comes with some risk. The first risk is being pregnant, because pregnancy tells us maternal physiology in various ways, which I talk about a little bit with you today. But there are other risks. For example, inherited disorders, fetal aneuploidies, chromosomal changes and deletions, and we touch a little bit on all of those. But there are other risks as well for early miscarriage, changes in fetal growth, and other diseases like preeclampsia and prematurity. So I'm going to walk you a little bit how the placenta plays a role there, what can be done, what can't be done, and where technology hopefully will go to improve pregnancy outcomes for patients in need. So what is pregnancy? Let's start really simple. It's by definition 9 months of growing a new life with the intent to the best prepared, be best prepared for the outside world. This is important because what really needs to happen, the baby has to know, the fetus has to know what it has to expect in terms of calorie intake, environmental factors to survive. Furthermore, as you might know as well, like evolution made human pregnancy very efficient. Although I'm going to talk about like various health problems, most of the time pregnancy goes very well and the outcomes are positive. But pregnancy is as well a stress test for the maternal physiology. So for women in particular, being pregnant is a huge challenge because due to the many changes of the physiology. It's not only weight gain and hormonal stresses. There are other factors that can impact maternal health. You probably heard about like gestational diabetes and gestational hypertension. So what happens there is we believe that the pregnancy itself pushes women who are already at a slight risk or increased risk for those disorders, hypertension and diabetes, over the edge to become diabetic and get hypertensive during pregnancy. Sometimes things can go wrong, and early detection of problems are needed to help patients. And our role in our research try to fill this gap. So what is a placenta? And you can see it on the right. It's beautiful, right? It looks like a pancake and with a middle inserted umbilical cord. You can see as well here on the top, this is the fetal side. If you're not familiar with placenta, but you will be very familiar at the end of this lecture, you can see here blood vessels on top. And they all end up in the umbilical cord, nourishing the fetus to ensure appropriate growth. It's a critical organ, but it's temporary. So that's why it's often poorly understood, because it's not well accessible during pregnancy. We don't want to poke pregnant women and cause problems there. It serves as a lung, as you can imagine, because it takes oxygen and transport this oxygen over the placenta to the fetus so it can grow, because the fetus is not breathing. It provides nutrients, all kinds of nutrients from transported into across the placenta to the fetus to ensure growth. And it removes waste as well, including gases that are not needed. One of the important aspects of the placenta is an endocrine organ. It releases hormones, but as well other molecules that can directly talk on the physiological level with the mother. So, for example, pregnancy-induced hypertension, we know that certain molecules can act on the endothelium of the mother, that is at the blood vessels, the lining of it, and can cause abnormal function that lead to hypertension. We see as well changes in the kidneys, for example, talk about it a little bit later, that resulted in dysfunction of the kidneys and increased release of protein into the urine, and we call this proteinuria. So all in all, the placenta is a molecular communicator between the mom and the baby. The placenta reads as well out certain hormones and the amount of calories or nutrients available to the baby. And by that, using this information and programs the baby, because it supposedly has to expect lower or certain changes in calorie intake when it is outside the womb. So by that, it affects immediate and long-term health of the mother and the baby. All in all, it carries information about the pregnancy status and its health. So how do we make a placenta? So as you all know, the maternal egg, or as well called an oocyte, and the sperm of the father work together to achieve fertilization. As we have now, we have two individuals creating a new individual who has supposedly grow in the mother. So where does this all happen? So if you're not very well knowledgeable about human reproduction, female reproduction is very, very well coordinated. It requires that the maternal brain and the hormones talk to the reproductive organs, and in return to ensure a cycle of what we call the reproductive cycle of creating an environment that's susceptible to pregnancy. And it starts here. We can look here a little bit. This is a schematic overview of the reproductive organ, the uterus. We have here the uterine cavity with the uterine lining, a cervical canal opening into the vagina. And on the side, we have what we call the ovaries. They produce oocytes, the eggs, in a fashion that is in a monthly fashion, as we call the reproductive cycles. As part of the cycle, oocytes will get released in the fallopian tube here, where if intercourse happens, the sperm would fertilize the egg. And then we see variety of changes in the nuclear, in the zygote, in the 2-cell stage up to the blastocyst, what we call the blastocyst. At this stage, the blastocyst will try to implant into the uterine cavity. And here's the lining, is the endometrium. The endometrium, as part of the reproductive cycle, at this point is most susceptible for implantation. This is the point when pregnancy starts and the placenta develops. Now let's have a look at the placenta for real. This is here's one. Again, you might remember the first picture I showed you. That was a nice washed one. This is reality. It's a pretty bloody business because the placenta is directly connected to the maternal blood. But it's as well very separated from maternal blood. And I show you how the communication between both the mother and the fetus work without mixing blood or the fetus, the maternal. So you can see here in the middle, the umbilical cord got clipped here. You see the baby is not here. And you can see here a little bit the fetal membrane. And you remember, see like here the big blood vessels, which extend all over. This is a very healthy placenta. An abnormal placenta could have cord insertion at the sides. It could be misshaped. Normally, a healthy placenta is round and with a central cord. So why is it so important? I mentioned earlier, it provides nutrients for the growing baby, and there is a communication going on that ensures about appropriate fetal development. And I mentioned before as well, it communicates to the baby what to expect outside the womb. So this is kind of important because what happens there is really the baby gets programmed by the mother what to expect outside the womb. And from this, actually, there is a whole research area, which we call developmental origins of health and disease, was built on this. It's based on the fact that, for example, changes in the womb will determine the risk for certain disorders later in life. There's an example for that. It's like it's basically the Becker hypothesis. It's called, originally described by Professor Becker in England, and he studied women during the war in the Netherlands, and those were exposed to very low calories as part of a famine, and those babies did get very low calories, but then they were fed higher calories after they were born, and those babies who had like low calorie intake before they were born, but then got high calories, developed a higher risk of heart disease and other disorders. So from that, a whole stream of research developed in like how to deal with changes in calorie intake and other environmental changes during pregnancy. The placenta can tell us a lot about the health of the fetus as well as pregnancy. I'm going to talk about this a little bit. So how does fetal maternal communication actually work? So you remember we talked about like here on top. Let me see if I can make my mouse up here. On the top again, you see cervix here. Here the implantation site on the right side, and here is a baby that grew in the cervix, in the uterus, my bad, in the uterus for a while, and you can see like how here you find the placenta, the umbilical cord, and the connection. Now, if we cut like through the placenta here, we get this here. You can see here the umbilical cord and the blood vessels that bring nourishment to the baby and remove waste in the other direction. Those blood vessels are covered, you can see here, with cells. We call them the trophoblast cells. Trophos is Greek means nourishment, and those cells are responsible for placental function. What you see here in the middle is all maternal blood. So the mother here has the endometrium, has structures, arteries and veins that release blood into what we call the intervillous space and give the placenta the opportunity to take up and release material into maternal bloodstream. As part of this, the blood will never mix, only molecules are crossed across a placental surface. So, so how does it work? To ensure that the baby can grow well, it works kind of like a parasite because it plants and tries to take over blood flow to the implantation site. This is the implantation site, to ensure that there is always a constant supply of nutrients to ensure appropriate growth. I want to highlight these structures here in the endometrium. They're called spiral arteries. These arteries release blood into this space. You can see the tree-like structures. We call them villus trees. And it's really like a tree that attempts to create a huge surface to be able to transport as many nutrients and gas as possible, to transport it through the blood vessels to the fetus and back. And here is probably the most complex diagram for today. I promise there will be nothing else like this. But if we now highlight this little area here, we see here how the placental tissue attaches here to the decidua that is the uterine wall. So the placenta anchors those cells and itself to the uterine wall to make sure it doesn't fall off. And while it is doing that, it as well transforms the blood vessels so the mother is unable to contract and control blood flow, because that will be bad. So we call this spiral artery remodeling. This happens in the first and second trimester, and it allows that the blood can flow constantly at a high oxygen level and sufficient to ensure the growing demand of nutrients during pregnancy. You can see here, like what normally controls the blood flow is because with muscle cells, and those muscle cells can contract, but the placental cells, the trophoblast cells that evade here the villus, we call them extravillus trophoblast cells, replace those cells and take over the blood flow. This is very important because in disorders, this often goes wrong. And you can probably imagine if blood flow is not high enough, there is a whole range of problems that can occur. And those that occur are pregnancy complications. You might have heard the hypertensive disorder preeclampsia. We have fetal growth restriction, where the fetus is not achieving the full size it's supposed to be as part of the pregnancy status and time, as well as preterm birth. Preterm birth is characterized as the child has to be born or is born before fully grown or the finish of the third trimester. And all of these are leading causes of perinatal mortality and morbidity. So there's a very intricate connection, and you probably already understood that from the last picture, between the development of this disorder that probably originated very early in pregnancy, first trimester. Yet we do not know how to identify the disease so early. And I explain a little bit why that is and how we may be able to overcome this. Often the only cure for a disorder such as preeclampsia is the delivery of the baby in the placenta. And you can imagine this in turn can have other effects and consequences, that the baby might be preterm delivered and or too small for the gestational age. And here maybe a picture that kind of summarizes it. You can see here a baby on the right that is of normal size. The baby in the middle is I have to look at the figure legend here. So this is like a really very preterm baby, small for gestational age, and a normal size. And you can see already visually, this is very striking. And you can imagine that this challenges for the fetus at birth as well as long term. but as well, like fetal growth has to be put into context, right? When we look at growth curves and growth charts, we have to consider as well that certain parents, if they are smaller, most likely will produce smaller children. So taking this all into context helps to see and evaluate the risk for these children. So how do we detect abnormal placentation? There hasn't been much changes over the last 40 years, and ultrasound is pretty much the tool of choice to measure form, structure, and blood flow that begins in the second trimester. As you can hear already, like first trimester is really hard to do, but it's a very important and functional tool. So I can show you a little bit here for our end users. You can see here blood flow of the arterial vein, and you see the pulsatility as well. Another way of looking into the placenta is, I talked about this earlier, that the placenta releases molecules into the maternal blood. And those molecules, if they are associated with a disease or with healthy placentation, we can measure them. We can measure them if they are in high enough concentrations and our detection mechanism is sensitive enough. You can imagine this is more challenging when you have a very small placenta as you have in the first trimester. And the sensitivity of those markers is not very high at the early stages. So overall, the inability to access the placenta itself, the tissue, means that the evaluation of placental function cannot really begin before the first trimester. We do not want to poke a pregnant woman to isolate placental tissue if it's not absolutely necessary. So there need to be a risk. Assessment made before we would ever do that. So generally, that is not being done, but it is being done when we look into genetic testing, and there is a significant risk. I'll talk about this a little bit later. So how is abnormal placentation managed and treated? So mostly it involves close monitoring of the high-risk pregnancies. And this is like accompanied-based bed rest, using ultrasound at various stages during pregnancy, and biomarkers. There are some treatments available. It's like aspirin is very common, heparin to increase blood flow, statins, and other drugs to prolong pregnancy to ensure that the mother and the fetus achieve the best outcome possible. But currently, treatments do not focus on placental function due to the lack of research and potential risks to the fetus. We want to avoid giving drugs to pregnant women naturally if we do not understand what the risk to the fetus is. But it's really hard to develop drugs in the first place because we have to give drugs to women who need them at some point. So there is like this risk-benefit problem, and it's important that we go through this, but as well like the lack of certain models, we test in mice, maybe in baboons and other animals, but at some point we have to help the women that really need it. And this is kind of where our research comes in. So how to access the placenta non-invasively. And now I talk a little bit about research we are doing or have been currently doing. And what I didn't tell you yet, but it's really important and it's pretty cool, is that actually placental cells can be found in the cervical canal. So by now you might remember, like this is the uterus here. Here would be on the left and right would be the fallopian tubes. You see here, and thus is this implantation on the top, the fundus, and the cervix would be here at the bottom. So if we look at this, what happens during pregnancy has been found in like 1970s, about like over 40 years ago, that certain cells, the trophoblast cells, that invade into the cervical, into the uterine lining, some of them escape and are naturally released through the cervix. Here's like a little bit another figure that kind of shows, like, remember the cells were invading, and some of them seem to be invading glands or outside the placenta, and then naturally make their way into the uterine cavity and then get released via the cervical canal. As you might remember as well, I talked about it a little bit earlier, showing you that these trophoblasts, placental cells, if they don't work very well, we don't get good spiral artery remodeling, we get affected of nutrients and other issues. And so the cells appear to be a good target to look at if we want to learn if there's something wrong with the placenta and the pregnancy. So our hypothesis was that cervical placental cells should carry information over the placenta and fetal genetics. Why fetal genetics? As you know, is like the placenta is not fetal, it's extra embryonic, but it carries similar genetic information. So these cells could provide information about the genetic status, but as well about the placenta. And I want to show you a little bit data that this is actually the case. So these cells are known for 40 years. My former boss worked on it as well, but it couldn't be really put to clinical use due to the lack of methods to isolate and analyze these cells. So how are we doing this, or how are we looking at those cells? If you're probably all familiar with a pap smear, and a pap smear is used in women to assess potential risk of cancerous cells in the cervical canal. It's commonly used as well during pregnancy, and it is deemed to be safe. So if we take a sample from a non-pregnant woman, and we look at the cells, and you can see here on the left, patient 5 and 6, we call them, you can see the classical cells found in the cervical canal. Then we took 2 specimens here from pregnant patients, and we labeled the cells with an antibody against HLA-G. HLA-G is a unique protein in the reproductive canal. Is only expressed by the trophoblast cells. As you can see here in brown, a couple of these cells are positive for HLA-G. And when we look at them, for example, for the Y chromosome, we can show they are actually of fetal origin. As you know, like boys have X and Y, and the mothers have obviously 2 X chromosomes. That is another way of showing that these cells are truly of fetal/placental origin. So now we know those cells are there. So how do we collect those cells? So what we do in our research, as early as 5 weeks of pregnancy, we are able to use a simple pap smear. Listen to a guy talking about a pap smear, to collect cells from the endocervical canal. It's a relatively fast procedure. It's sometimes accompanied with the first perinatal visit. It's a fast procedure, and then the samples get preserved. We rinse them in a transport medium, and then we send them to the laboratory. And in the laboratory, we do something that's pretty cool. Remember the molecule HLA-G? So it is a protein, right? It's expressed on the cells, it's present. And then there are molecules called antibodies that can detect certain proteins. So here we have an antibody against the protein HLA-G. That means this antibody would bind to the HLA-G. Now we couple the antibody with a particle that is magnetic. We call it magnetic nanoparticles with anti-HLA-G. We mix those together with the cells, and the nanoparticles with the HLA-G will only bind to the trophoblast cells. And then we use a simple magnet, we pull the cells out, and then we can do pretty much what we want with the cells. One idea is looking at the DNA, and or you use the cells for other analytics. So what are the applications for those placental cells isolated from the cervix? As I mentioned, we can identify genetic abnormalities, and we can identify placental health. I'm going to show you 2 or 3 examples here. So I'm not sure if you're not in the field of prenatal testing or screening, I'll give you a quick overview. So the most invasive ones, you see like a nice long syringe here, is chorionic villus sampling. What it does, it is a needle that it gets placed into the placenta, and we take a piece of that and we take it out and then we can look at the DNA in very great detail. It enables full genetic analysis, very important. And as well, like for compared to non-invasive, it does not provide, it has really a lot of the fetal DNA and only very little contaminating DNA, but it is very invasive. And there's another challenge. The placenta is can be a little bit different than to the fetal DNA. It's called mosaicism, and that has to be considered if these tests are done. Then there's a second test, it's called amniocentesis, and there the amniocytes are collected. And you can see it is even a little bit more challenging, although it's pretty safe in trained hands, but like the risk is real. And there the amniocytes get collected from the amniotic fluid. So all of these look very invasive, right? And they can be done at 10 to 15 weeks and 15 to 20 weeks. And then the gold standard, or one of the most used assay currently, is non-invasive pyramid testing. As you maybe remember, like from my earlier slides, the placenta releases all kinds of stuff into the maternal blood. It just like gets sloughed off, right? So placental fragments and placental cells get released into the maternal circulation. Remember, as a blood flow and implantation site, it breaks off, is there, and as well the cells break apart, and fetal DNA to a certain concentration can be found in the maternal DNA. So what happens, you can take a few milliliters of maternal blood, and then you can isolate what we call cell-free floating DNA, fetal DNA. And then you can do analytics of it. But there are a couple of challenges with that, because there is a huge amount of maternal DNA as well present. So you have to separate that information from the fetal information. And that can be very challenging, because there are maternal factors like body mass index and others, as well as gestational age, that can cause challenges in determining the genetic status of the fetus. Normally, what we look in those fetuses for very big changes. You probably are all familiar with trisomies. That means there is a third or second chromosome that shouldn't be there in the fetus present, and we can test for this pretty easily because we have a lot of data. It is hard or impossible at this stage to look for very, very small deletions. For example, as you find, we call them single gene disorders in sickle cell anemia, where we have a point mutation that can cause a disorder. So there are big advantages to this technology. That's why it's so widely used, but as well, there are significant limitations as well. So our group then in 2016 investigated the potential of the trophoblast cells to enable genetic testing for fetuses as early as 5 weeks from a simple pap smear. So what we did is we had like 20 patients or 25 patients from 5 to 20 weeks of pregnancy. We took swabs from them. We had as well a piece of the DNA of the placenta itself. And what we did is what we call fingerprinting of the DNA. Fingerprint is we look for certain markers in the DNA. And you might remember us from CSI. We find DNA markers that can distinguish the mother from the fetus and the father. And what we did then, we compared the DNA from the pap smear to the placenta, which was identical, and it was different from the mother to 50% and different from the father to 50%, exactly how it's supposed to be. And those DNA markers are, I think we used about like 100 markers, and it was very easy to precisely pinpoint the differences in the fetal genome. This got a lot of attention because it provides new means to now look very early and very precisely if you get those cells into the genetic status of the fetus, which will help for treatments, for management of disease, has a lot of implications, obviously. Now I want to switch to identification of placental problems in the first trimester. So as you can imagine, because genetics is so much done, like I'm looking, although my background is not in genetics, it was a low-hanging fruit to look into the DNA of those placental cells. But my research interest is really into placental development, into the underlying mechanism of disease. When I heard about those cells, I was like, amazing. We can now actually, in the first, because we were all relying on placental tissue to investigate after, obviously, the placenta was giving birth, right? So, but as in cancer, that is like the disease is so far ahead, it's really hard to pinpoint what originally caused the problem. So, as you can imagine now, looking at cells that are responsible in the placenta for function and disease provides us a new window into pregnancy. Can identify high-risk pregnancies, exclude low-risk pregnancies is important. If you can tell somebody, you know what, like you're a high-risk patient, but it looks like you're doing just fine, and it can direct management and develop targeted treatments. Because getting preeclampsia very late, or getting preeclampsia is like getting a cough. There are a lot of ways to get the disorders. Some are rooted into the placenta, but if you have malaria or other disorders, you can get symptoms that are similar to preeclampsia. So it is really important to know what caused the disorders very early. So what we did was we did 2 pilot studies, and I just like walk you through it very briefly. We took those cells from pregnant women. We had like a library of them, and then we picked healthy outcomes. Once we knew how the pregnancy went, we looked at early pregnancy loss, pregnancy that didn't go to term, and we looked as well for patients that had disorders such as preeclampsia, the hypertensive disorder, or intrauterine growth restriction, small for gestational age. We took those patients and sample, then we took the slides out of a freezer, and then we did the following. We looked for molecules that we knew were associated later in pregnancy in the blood, and at term, when we look at the placenta, with abnormal placentations. And then we took the cells and looked for if we could see changes in the cells in the first trimester before any clinical symptoms occurred. If we could already see changes, and in fact, we were able to. So we looked at certain proteins, like 6 biomarkers. I will not go into the details. We used the staining method I showed you earlier, and used like a semi-quantitative ways to quantify proteins in there. We fingerprinted them, we compared them, let the computer group the different samples. And what we saw that the abnormal pregnancy nicely separated from the data of the healthy, indicating that they're, number one, the trophoblast cells in the cervical canal have very early in pregnancy already changes we can detect that might be indicative for later disorders in pregnancy. This pilot studies are now the foundation for other studies we're doing to develop clinical tests very early to ensure that pregnancies can be measured and successful. So 38 minutes, good in time. In summary, I hope I could show you the placenta is pretty awesome and mysterious. It carries information on the presence and the future of fetal and maternal health, not only pregnancies, but as well beyond. It can inform on genetic and pregnancy health, and more research will be needed to develop new development into clinical tools. I want to thank you for listening tonight and acknowledge the many, many people I had the pleasure to work with here at MSU, Spectrum Health, Mayo Clinic, Wayne State, and around the world. If you have any questions, happy to take them. I think some of them will be found in the Q&A. Thank you very much. I hope you had a good time.
All right. Thank you, Dr. Drewlo. And it does seem like you've piqued the interest of a few people out there. If you have questions, feel free to use the Q&A section, which is just at the bottom of your Zoom tool, or maybe elsewhere, depending on the platform you're operating on. So we do have a couple here. I'll go ahead and read a few. So this person writes that they have worked in countries with much higher rates of eclampsia than the US. A number of years ago, there was some question that calcium deficiency could be a risk factor. Studies of calcium supplementation had no results, but may have been done in women primarily with calcium-rich diets. Has anything more been done more recently in populations with low calcium levels?
I would not have the answer to that, sorry.
Gotcha, okay. Maybe a little bit related. About 15 years ago, there was a few small studies noting a reduced incidence of eclampsia in high-risk women who had taken macrolide antibiotics for unrelated indications. The observational study was from South America and a follow-up small randomized controlled study in Bangladesh with impressive results. Are you aware that any more has been done with this?
So no, I am not. I think I would have heard like I haven't heard anything in the high risks scene, like really like when I heard calcium earlier, calcium magnesium was one of the treatments that have been tested, but the studies are really, how you call it, inconclusive often, and you will hear this with many of the studies, and it relates to the fact that the disease spectrum is so diverse, and if the data is not stratified or big enough, it is really hard to find positive results.
Thank you. In cases of multiple gestations, are there still ways to use the pap smear collected placental material?
It's a fantastic question. We haven't done it, but technically, absolutely yes. Bioinformatics wise, it should not be a problem. But it's a great question. I love it.
Yes, these are all my questions, by the way.
Well, there you go.
All right. Is the patient required to prepare her body or cervix for the trophoblast cell pap smear?
Nope.
And so what's the clinical process?
So a clinical process normally depends on where we do that. So in general, we don't want to interfere with clinical practice because that is always easier, as you know, to get successful patient enrolled. So it's normally as the first perinatal visit, when there's an ultrasound, we suggest that that the sample is taken first before like an ultrasound probe, if it's transvaginal, is used. But beyond that, that is pretty much it.
Great.
It's very simple, very straightforward.
Does having polycystic ovary syndrome affect placental health?
I can't comment, but I don't know.
Here's another one. Are the materials needed for this analysis considered high tech, or are they available in low income areas?
So I would call a pap smear very low tech. I think it depends then on the follow-up analytics, and I would assume like sequencing is becoming more common. I think you will need an urban center, and like if you look into genetics, that would be my answer.
Okay. Here's a question from a teacher here at Alma. My students know I love the placenta. It's a femtomaternal origin. Are there maternal alleles known to make given women more likely to contribute to a healthy placenta?
It's the holy grail of information if we would know that. But we know that there's certain genes or areas in the placental genome, which comes from the mother and the father, can contribute or increase risk. We do not fully understand the development, the origin and the development of the disorders. And that has probably, for those people who are a little bit into genetics, to do with epigenetic changes in the placenta.
I have one last question here. So if there are any others out there that would like to ask a question, please feel free to use that Q&A section. This is from an aspiring doctor herself who's just starting her undergraduate years at Alma College. And she would like to know, what is your biggest piece of advice for someone who is interested in becoming an OB?
So I am not a medical doctor, so I'm a little bit biased, right? So I think but I have people I look up to very much, and I think integrating clinical and medical education with research, I think adds an extra valuable layer to become a successful doctor. So of course you have to do research.
Well, I'm running thin on questions. I'm not finding any more here. We can wait a couple moments and see if a few more chime in. Here's one. I've heard this myself before, and this person admits this is a strange question. Are there proven
I was waiting for that.
Are there proven benefits to consuming the placenta after giving birth?
No, there are proven benefits. There are proven problems when you consume the placenta, and that is probably an infection you get, and you might give it to your baby. There are anecdotal stories about like postpartum depression and others, but there is no scientific evidence that this would be beneficial. So please don't eat it. Grow a tree.
Doesn't sound very appetizing. Are there non-invasive techniques replacing routine amniocentesis?
Yes, I hope the one I just presented.
Right.
But it's an important question because as Tim just asked, because there is the amniocentesis is really the ultimate test, right? Because it is true of fetal origin, where is the placenta is extra embryonic, and there is a little bit difference. So either no or yes.
All right. Well, that was our final question. And last time I said that, we had a couple more pop-ins. So we'll see. Well, maybe that brings us to an end here. I think our questions have ended, Dr. Drewlo. I think we'll go ahead and close our session today, and I would like to thank Dr. Drewlo and everybody else that made this possible, and all of you, of course, for attending today. Thank you so much.
Thanks, everybody. Thank you so much, Dr. Turk, for moderating and inviting me. Much appreciated, and thanks for listening in tonight. Have a great evening.
And you're welcome. And good evening, everybody.
Bye-bye.